Sir, you can start now. Sir, you can start now.
But there is some disturbance, it seems. Okay. Namaste to everyone. I welcome you all for second day of 3-day webinar. And I would like to introduce today's speaker, Dr. K. Chandrasekhar, who has already joined with us. And I'm very happy to share with you all that he is known to me since his B.Pharm days. He was 2 years junior to me. He has completed his B Pharmacy in the year 1997 from Kakatiya University, Warangal. So 93 batch. And immediately after completing B Pharmacy, he has studied M Pharmacy also in Kakatiya University, and he has completed it in the year 2000. And subsequently, he has pursued his PhD also in the same university. And it's surprising to see that he, starting from B Pharmacy until his PhD, he has continuously, without any break in the middle, he has studied his courses in Kakatiya University itself, which is not very frequent and which is very rare. And he was a gold medalist in B Pharmacy level in the university. He has received gold medal for his performance as a student in the year 1997, and he has received a fellowship JRF and SRF for his M Pharmacy and PhD work. And later he has moved to US to pursue his postdoctorate, which he has successfully completed in the year 2008. And then onwards, he worked in academics. Presently, he is working for California Health University as a professor. And he is overseeing 2 research products which are funded by US government. He has rich experience in the field of academics, starting from his student days. So when you keenly observe his biodata, one can easily understand that he has associated with Kakatiya University for quite long time, from B.Pharm days till his completion of PhD, almost 93 to 2008 means 2004 means 11 years. And once again, you can see his association with California Health University is also long. That is almost equal to 7 years. So which shows that he's stable and he stuck to one university and where he has excelled to the max. Personally speaking, for myself, I'm very lucky to have him as today's speaker, and he's a wonderful gentleman. In addition to this, he has got so many publications to his credit, and he has co-authored some books. And for full details, kindly refer to the file which I have circulated in our WhatsApp group. With these few words, now I would like to request today's speaker, Dr. Chandrasekhar, to kindly take up the session and enlighten the viewers. Thank you. Chandrasekhar, please, sir, you can please go ahead.
Good afternoon, everyone. Thank you so much for the nice introduction, sir. I'm not sure if I deserve so much praise, but I'm proud to say that I'm a product of Kakatiya University, and Ramana sir was our senior, and I'm blessed to have such a nice seniors Okay, they all led my way to my success. Okay, I shall ever be thankful for them. Okay, yeah, I will not take much of time in introduction, but let's briefly discuss my research area. Okay, I would like to give a short, brief introduction about myself. That is like once I moved to USA, I did my postdoctoral research in transdermal delivery in Dr. Ajay Banga's lab. Following 4 years of postdoctoral research, I joined California North State University as an assistant professor, and I was there for about 6 years. Then I moved to California Health Sciences University in California, and I was there for 7 years. There I grew from associate to full professor. Right now I'm working as a professor in pharmaceutical and biomedical sciences, responsible for teaching calculations, pharmacokinetics, pharmaceutics. Okay, my primary area of research is transdermal delivery, and I'm going to share a project that I did in transdermal delivery, which has some scope and applications. And this is not a difficult project to do in India, so that's the reason I chose to present this to you all. No fancy equipment is required, as small micro molding machines, maybe.
Chandra, can you please share the
uh,
screen if
yeah,
there is some.
sure.
Sir should say share, sir.
Yeah,
Yeah.
that's what.
Can you see?
Yes, yes.
So I'll be giving a talk on microneedle mediated transdermal delivery. Transdermal delivery is nothing but delivering the drugs through the skin. Why we have to use skin? Like we know that like oral route is the most commonly and very widely used route for delivering the drugs. Almost 9 out of 10, that means approximately 90% of the drugs are delivered by oral route. But there are certain drugs that cannot be delivered successfully through oral route. For such things, we have to go for alternate routes, and transdermal delivery is such an excellent alternative. It is because it is the largest organ and readily accessible. Like you can, there is no special techniques required, and it is a huge surface area of about 16 square feet. And also, especially in the Western world where there is less suffocation and sweat, there is a huge potential for its market. There are several products in the market in the Western world which are transdermal, especially nicotine, smoking cessation, pain patches, and all that stuff. Okay, therefore it has a huge potential. And certainly there are certain advantages for this transdermal delivery. It is painless, unlike injections and all, it is painless. It doesn't require any invasive kind of stuff. And as compared to the oral route, it bypasses the hepatic first pass metabolism. As you know, drugs get degraded by first pass metabolism when given by oral route. So this transdermal delivery is an excellent alternative to bypass this kind of first pass metabolism. Protein, peptide, any kind of drugs can be delivered using some active strategies. And it reduces side effects such as gastric irritation. All those side effects can be reduced. There is more better patient compliance because it doesn't have to take a medicine. It is usually once a week, once a 3 days patch. So just stick the patch onto your skin and you're good for 3 days. So there is no need of taking tablets 3 times a day, 4 times a day, or you don't have to swallow big pills on regular basis. The beauty of this is like it provides a prolonged duration of action. Some patches can provide as long as 7 days. One patch is good for 7 days. There are several antihypertensive patches, that is for controlling the blood pressure, that can deliver the medication for about 7 days. And another versatility with this is ease of termination. Say, for example, if you take a tablet, and the patient is feeling side effects. There's nothing you can do because already the drug is in the system and you have to wait until the effects are gone. Whereas in this case of transdermal systems, once you see the side effects, you can immediately remove the patch and you can terminate the side effects. That is one major advantage with this transdermal systems. Can you guys hear me? Is this clear?
Yes, sir, you can carry on.
Okay, thank you so much. So, however, despite these several advantages, there are certain limitations with this transdermal delivery. The main problem is it is only applicable for lipophilic molecules so far. Okay, so mainly lipophilic molecules can be delivered using. If the drug is hydrophilic, it cannot bypass the lipidic nature of the stratum corneum on the skin. So it is primarily limited for lipophilic molecules. And molecular weight is typically less than 500 daltons. And very low dose. You cannot practically put more than 20 milligrams in a patch. Typically, they are about 10 milligrams in a patch. So if you want to deliver grams of drug, certainly this is not a good strategy. And also certain drugs that are highly potent are given through this route. So if you are giving certain drugs like nicotine or something which is highly potent, and if your pets happen to chew that patch, then there is a danger of like risking its life. So you have to be careful with that. So if you look at the basic structure of the skin, skin can be basically divided into 3 layers. The uppermost layer, we call it as an epidermis, the dermis, and the subcutaneous layer. We are primarily concerned with the epidermis. Okay, it is a superficial layer, but if you see on the superficial layer itself, there is another top layer. So what is going to happen is this, the superficial layer, then that superficial layer is about 10 microns in thickness. When I say 10 microns, it is less than the thickness of a newspaper. Okay, but this is the most important protective barrier for your skin. It is the one that offers the primary line of defense against entry of substances. Every dirt, water, anything, it prevents. It is primarily lipophilic in nature and is made of dead cells. So practically resistant to everything. So this is kind of boon given to us from microbial attack. But the same thing can limit the delivery of molecules. So what we are trying to do is, we are trying to manipulate the superficial layer to some extent. Okay, we are going to breach or manipulate the manipulate the superficial layer, and we reduce the resistance of the superficial layer and then deliver the molecules temporarily. That is how we do using the microneedles. I'll discuss this in later slides. Okay. Now, if we take directly the plain skin, the delivery of molecules is very limited, especially if the skin is intact, then you can, your choice of drugs is very limited. Very highly lipophilic molecules and less than 500 daltons can be delivered. However, we can increase, that means we can enhance the delivery using techniques such as physical enhancement techniques and chemical enhancers. I'm not going to go into the details of chemical enhancers because you might be knowing terpenes and all that stuff, surfactants, terpenes and all that. There are plenty of chemical enhancers. In case of physical enhancement techniques, we'll be using the ultrasound, antiphoresis, electroporation, radiofrequency ablation, laser ablation, thermal microporation, and microneedles. Among these, I have worked on ultrasound, antiphoresis, electroporation, thermal microporation, and microneedles for delivering the drugs across the skin. However, I'll be limiting my lecture to microneedles. So now microneedles. What are these microneedles? As I said, microneedles. It is a needle, very small, tiny, very tiny microneedle. This is a small, as small as, I'll show you in the next picture. It is arranged in the form of a layers, in the form of a specific designs of microneedles. There will be multiple microneedles sticking to a patch. So if you want to look into the size of the microneedle, see on the top right picture, okay, here, the top right picture, this is the size of the needle. The bigger one is the size of a needle, that is a subcutaneous injection needle, tip of the needle. These are the scanning electron micrograph pictures, okay? And as compared to the tip of the injection needle, these are the microneedles. These are tiny, as you can see, these are very tiny, very small, okay? And they are pyramidal in shape. You can see from the bottom picture, they are kind of half-cut pyramids. So if you zoom further, the tip of the microneedle looks like this. Entire tip of the microneedle looks like this. And it is about 3 micrometers in diameter. The entire circle, the tip is about 3 micrometers in diameter. When we insert in the skin, this is an intact stratum corneum. When we insert in the skin, depending on the design, like it forms a hole through the stratum corneum. There is a hole through the stratum corneum. And this hole, that means once we create or breach the stratum corneum, it is the skin becomes more accessible. It becomes more permeable and more accessible. Okay. Now, how do we use these microneedles? Each of these microneedles are arranged in layers. You stack them in piles, okay? They get attached to a base, okay? You push the base onto the, you apply slight pressure. As much as the pressure required is just like approximately equal to that of pressing the button of an elevator, that is pressing the button of a lift, okay? So once you stick it to your skin, the microneedles are going to enter, okay? And because these are made of soluble, materials such as maltose, there is some amount of water inside the skin. This water is going to dissolve these microneedles, release the drug, and the base is gone. Base is removed. So microneedles remains, the maltose part remains within the skin. So maltose is, you may ask, is it toxic? No, it is generally regarded as a safe substance. As you know, GRAS category of substances. It is safe, sterile, and biocompatible. So there is no problem as of using this microneedles. Another type of needles that we use are the solid metallic microneedles. We disinfect them before use. These are available as a cheap alternative. These are arranged from different sizes. And typically we use 0.5 micron millimeters, the first one, 0.5 millimeter size one. So these are arranged onto a roller like this. And to show the magnified picture, this is like this. It's about 500 micrometers tall. And the tip looks like this. The scanning electron microscope from tip, it looks like this, okay? The advantage with this is these are reusable and can be used for multiple applications. And each time you can sterilize them using alcohol or something, okay? So when they are treated on the skin, you can see this is on the left side, right side, you can see the stratum corneum, the skin is breached. you can see a clear hole with the piercing of these microneedles. Okay, that is where the stratum corneum is damaged. Okay. So this is a normal skin. It's a staining. Here you can see the upper layer, which is a stratum corneum, breached by microneedles. H&E stain of the skin that shows clear breaching of the stratum corneum. Now, I want you to understand, when I say breach, this is only to the top layer. It is not going deep inside, because the microneedles are so short that they will just breach the superficial layer, but it will not go into the deeper layers. The problem is, once they go into the deeper layers, you encounter these nerves and microvasculature, and there is high chances of getting infection. The superficial layers of the skin, there are no there are no microvasculature. There is very limited microvasculature, and the odds of encountering them are very, very low. So you won't feel pain, number one. There won't be any chances of infection, number two. That is the advantage with them. And the third advantage is, this kind of damage, even if it is caused, it can get repaired within 24 hours. So the moment I remove the patch, in the next 24 hours, skin becomes normal. So it is like, that's the most important advantage with this. Yeah, the holes are not going to remain forever, but the moment the patch is removed, they are going to close. I have data supporting that one, which we'll be seeing in the later part. Okay, we call it as, the method that we use, we call it as poke and patch method. It is like, the moment we call it as poke and patch, that means like we poke the skin, let it stay there for a couple of minutes, then we remove the needles. Okay, the drug, then we place a patch so that the drug is released from the patch in a controlled manner. Now, there are three basic aims for this my project. One is, I want to see if these microneedles are able to penetrate and create micropores in the skin. That is my first aim. That is, I want to characterize these microneedles for their ability to form the micropores. And second thing is, even after forming these pores, are they able to increase the delivery of drug molecules? I want to check them in vitro and in vivo. Okay, or whether there is microchannels formed have the ability to increase the delivery in vitro and in vivo. And the third thing is, most important, I cannot leave this open, right? Once the channels are created, I cannot leave this open. I have to repair them. So I want to see how long it takes for the channels to close back. So there are three main aims. The channels are created, the channels are able to deliver the drug, and the channels are able to close once I'm done with my delivery. Okay. Let us see a supporting data for each of these. So characterization of micropores. See here, I have taken, I use the dye binding studies. These dye binding studies is nothing but, I used a hydrophilic dye, methylene blue. So using a hairless rat skin. Under normal conditions, rat skin is impermeable. Okay. Once I apply the methylene blue, it gets washed away because it is like it doesn't take any stain. But once I treat the skin with microneedles, what happens is the superficial layers of the skin is damaged and channels are created. When I say channels, these are nothing but the pores. Tiny pores are created in the skin. And inside the skin, it is all hydrophilic environment. So methylene blue dye diffuses inside. So wherever there are channels, methylene blue dye gets stained. So you can see on the left side picture a nice array of micropores. The same micropores, if you see a magnified vision on the right side, you can see diffusion of the dye within the micropores. Okay, so this is how these micropores are created. By using these dye binding studies, I can say that these micropores are created. Okay, give me a second. The next method we use is, say for example, all of us, as you know, in summer, there is a lot of loss of water vapor through the skin in the form of sweat. So whether it is summer, winter, day, night, whatever it may be, there is certain degree of water loss through our skin. Always water will be evaporating through the skin. So we call it as transepidermal water loss. Okay, that is the water that we lose in a period of time over a certain area of the skin. We call it as transepidermal water loss. You can see the right side top picture. Okay, here it is a normal skin. Normal skin loses water to a limited extent, very small, in small quantities. But the bottom picture, the bottom picture, once it is breached, that is once the microchannels are created, what happens is water vapor tends to evaporate at a very higher rates. That is, the loss of water will be increased. So we took this advantage of this water loss as a measure to study the permeation or study the extent of damage across the skin. You can see here on the left side, there are bar graphs. As a controls, what we used is hypodermic needle with 2 pricks, hypodermic needle with 8 pricks, hypodermal needle with 16 pricks, hypodermic needle with 25 pricks on the skin. So once I increase the number of pricks on the skin, the amount of water vapor is going to increase. At the same time, using microneedles, 1 layer of microneedles, 2 layers of microneedles, and 3 layers of microneedles. As I increase the number of layers of microneedles, water vapor is steadily increasing. Okay, and this is the base of the microneedles. The control, we use this base of the microneedles as a control. When I say base of the microneedles, it is nothing but it's a microneedle, but without microneedles. We have removed all the microneedles, we just pressed on the skin, that serves as a control. And this is an absolute condition on the right hand side where we tape strip, that is entire stratum corneum is taken off. Entire stratum corneum is removed, all the defense barrier is removed. We strip off the stratum corneum and that results in a tremendous increase in the water loss. Okay, by using these studies, like you can see that with using the microneedles, certainly there is increase in the permeation, water permeation across the skin. Therefore, we again confirm that the microneedles are able to form channels across the skin. The next one, what we did is, we want to see, now, as you know, like I have taken the microneedles, I've created pores in the skin. I have confirmed that the pores are there in the skin, okay? Now, I want to see how deep they are going. I don't want them to go too deep. At the same time, I don't want them to be too shallow. So I need to have optimum depth. What will happen if they go too deep? They're going to encounter this vasculature. Once they encounter the vasculature, what is going to happen is, it is going to cause, it is going to go into the bloodstreams and cause infections. Or there will be some sensory nerves in the dermis. So you don't want to prick them, that will perceive pain. So the best method to see the depth of formation is a confocal microscopy. Using confocal microscopy, I applied fluorescein, and then like once I applied fluorescein, I started imaging different layers across the skin. So this is at 20 micron depth. There is almost no fluorescence. And this is at 60 micron depth. There is fluorescence. 80 micron depth, there is fluorescence. 100 micron depth, still fluorescence. And starting at 120 micron, it started fading away. And 160 microns, practically there is nothing, no fluorescence, almost gone. So I can say that like the microneedles have permeated approximately about 150 to 160 microns. Okay, that is exactly the depth we wanted to see. Something between 150 to 200. We just want to stop less than 200 microns. Okay, these were successfully able to penetrate to that extent. So the depth of penetration is optimum so that it is not hitting the blood vessels or the sensory neurons. So the next study is like, okay, I have created the microneedles, they are there in the skin and they're going sufficiently depth. How uniform each of these microneedles take up the stain? I want them to take up like uniformly, relatively uniformly. 100% uniformity is not at all possible, but like we will see how much it is. So actually, these are the 6-layer microneedles applied on the skin and applied calcium imaging. This is like applied calcium. Then immediately took a picture and processed this image using MATLAB for the intensity. And you can see on the right hand side, you can see there's a pretty much uniform distribution. It is like the majority of them are between 1 to 4 permeability index. We call it as pore permeability index. This is a measure of the flux of these microneedles. So we have relatively uniform flux as compared to, most of the delivery methods. Okay, having said that, like now I was able to establish creation of pores. Second is the depth of permeation and uniformity of these pores. Okay, now my aim is, what is this practical applicability? Okay, how are they applicable to drug delivery? So this I have studied using the France diffusion cells, especially the in vitro experiments. It consists of a chamber. I think whoever is working on transdermals know. It is consists of a chamber. Is a chamber. We call it as a donor chamber, receptor chamber, and clamped. This is a membrane that will be clamped between the donor and the receptor. This will be connected to a circulating heater jacket. And the front part is a sampling port where we pick up the samples at intervals. You can see on the right side, I use the nicardipine hydrochloride as a modal drug. This is the passive transdermal delivery. At the base, you see is the passive transdermal delivery. And you can see that using the microneedles, there is a significant increase in the transdermal delivery. So this is the graph that shows a tremendous increase in the delivery of nicardipine hydrochloride in vitro. The same thing we did for in vivo. We want to continue the experiments, and I'm not going to details of this in vivo experiments, but this is how the passive delivery looks like, and this is how the in vivo data looks like. And we conducted this study in hairless rats and in Dr. Ajay Banga's lab at Mercer University. And data was very pleasing. And then next aim is like, these are like nicardipine is less than 500 microdaltons. Then later we want to investigate, is this method feasible really? How feasible is this? So The main problem, as you know, for transdermal delivery is its molecular weight. So anything less than 500 daltons can be delivered, no problem. How about, because we are creating the pores, so can we deliver large size molecules? We went on increasing the size of molecules, and we went up to delivering human IgG, which is about 130 kilodaltons per day. So we were successfully able to deliver human IgG in vitro, and it is about, this is a profile. This was, flux was several fold higher. And we were able to get about $300,000 grant from Pfizer basing on this data. And our next aim was, it's good. The microneedles are working. Microneedles are working, and they're able to deliver drugs more as compared to the passive. And so now we want to see, how about we use combination of enhancement strategies, like antiphoresis combined with microneedles. So that was one of our aim. So this is how it is, like a small electric circuit. Once you connect, if the drug is placed under the positive charge, the positive repels the positive molecules, and they are pushed into the deeper tissues. Okay. So now our aim is like, okay, we treat the skin, we use microneedles, we use, on top of microneedles, we use antiphoresis. That is not one enhancement strategy, but more than one, that is two enhancement strategies, microneedles and antiphoresis. Okay, as you see from this graph, the bottom layers, the lower layers are, the lower layers are the ones that are simple microneedles, base and microneedles. And the middle one is the antiphoresis one. And upon combination of microneedles and antiphoresis, it is much higher. You can see the top graphs. Microneedles plus antiphoresis is further higher. This is really a great, finding for us, and we did publish several articles on these combination strategies. So microneedles in combination with antiphoresis was able to deliver much higher loads of these drugs. And another beauty is it was able to deliver even the large molecular weight substances more efficiently. Okay, for these experiments, we use the prochlorperazine edicillate. Okay. This, as I said, like we were able to develop large molecular weight substances. This is a classic example, heparin. Heparin is low molecular weight heparin, and excuse me. This we have tried delivering in vitro. You can see passive delivery, almost negligible. We could hardly find the levels in the receptor cell. With antiphoresis, there was some. With ultrasound, there was some. With microneedles, there was some. All these three were the enhancement strategies. I mean, even microneedles was much higher. But like when I combined these microneedles with antiphoresis, it was significantly higher, way above. So this is one nice finding that we presented in International Journal of Pharmaceutics. So using the same method, we want to try this another anti-inflammatory tripeptide, so that we want to see the versatility of these microneedles. Can they be used to deliver the hydrophilic peptides? So we use a tripeptide kPv, and this is the microneedles, the purple line. The blue line is the antiphoresis. And you can see there is a huge delivery using antiphoresis plus microneedles. We are able to deliver this tripeptide. It is a kind of anti-inflammatory tripeptide investigational. So that is it. Next one is using the same thing. We want to see what kind of drugs can be delivered efficiently, because we are able to deliver high molecular weight, we were able to deliver low molecular weight. So we want to see, does hydrophilicity is going to matter? So we took a series of beta blockers, propranolol, acebutolol, atenolol, and sotalol. All of them have the same pKa range, and so they tend to get ionized to the same extent. So using this one, we tried to deliver using antiphoresis and microneedles combination. And it was found that like highly soluble sotalol and atenolol are able to deliver, we were able to deliver higher levels of drug using antiphoresis and microneedles. So later we use this technology for different modifications, different drugs. We use these methods for delivering the buccal delivery. Everything was great. I mean, like they were good. And one thing that you may be concerned is like, what would be the cost of these microneedles? Earlier, when we started, it was expensive, truly expensive. And it was about $40 per piece. But he used to offer us free because we were sharing the data with him. But now it has come to like 1 cent per microneedle. So it is, it is very doable and it is very easy now, right now, to work on these microneedles. So ultimately, I have created the microneedles. I was able to show that the channels were formed, the pores were uniform, and I was able to deliver the drugs in vivo. I was able to deliver the drugs in vitro. I was able to demonstrate that like different kinds of drugs, different molecular weights, whether it be normal drug, whether it be protein, can be delivered. And I was able to show that like it can be used in conjunction with other enhancement strategies. But the ultimate aim, where you get, where the critical factor is, I should be able to show that once these spores are created, they should close. What if they don't close? Yes, it becomes an issue. If they don't close, then it is very likely that you might get infected. So FDA is no way it is going to accept this if the pores are going to remain as is. Now we wanted to study what is the recovery time of the pores. So what we did is like we measured again the transepidermal water loss. As I said, like the loss of water across the skin. So we poked the skin and left the patch like that. Okay, removed the patch, occluded it for some time, and we removed the patch. Immediately after the operation, you can see tremendous increase in the evaporation. And after 5 minutes, 10 minutes, 15 minutes, 30 minutes, 45 minutes, 1 hour, 2 hours, 3 hours, 4 hours, it is going to decrease. It has decreased significantly. It has almost reached a passive part. So in approximately 2 hours, there is no pores present. And also we stained them later. And once we stain it, like there is no stains, like the skin is not taking up any stains. So it means that like we were able to complete this successfully. We were able to demonstrate the formation and closure of the pores. Okay. Another study what we did is like calcium binding pore closure studies. This is a normal skin. We poked the skin and applied calcium. That is immediately. After 4 hours, you can see that the drug is getting diffused. After 8 hours, you can see the drug is further getting diffused. After 12 hours, the diffusion is gone and the skin is becoming normal. In vivo, this is in vivo. So after 15 hours, pretty much the skin looks normal as compared to the first one. So we were able to demonstrate using these pore closure studies that these pores are able to close successfully. Okay. So these are the conclusions of my experiments. Microneedles have the potential to broaden the horizon of transdermal delivery. When I say potential, the broaden the horizon of transdermal delivery means using microneedles, I am able to deliver even the hydrophilic drugs. Otherwise, it was not possible earlier. Only hydrophobic drugs used to be delivered. But now I can deliver hydrophilic drugs as well. And second thing is small molecules, any type of molecule. Give me a molecule, I can put through the skin. Whether hydrophilics, macromolecules, vaccines, whatever it can be, I can put through the skin. That is one versatility with these microneedles, especially when used in conjunction with the other techniques. I have used ultrasound also for delivering these vaccines along the microneedles. They were really good experiments. Like it was like the data was real good. And these tiny microneedles, they're so beautiful. I myself volunteered and several of us volunteered. These can bypass a skin and no pain there is. The only thing that you feel is a slight pressure when you apply. That's it. You don't even feel the pain, nothing. No infection, no bleeding, nothing across the skin. And another beauty of these microneedles is these microchannels can be used in conjunction. You can apply on the microchannels antiphoresis or ultrasound or any other methods that can enhance the transdermal flux. Okay, and you can increase the patient compliance because you can reduce the patch size, you can decrease the dose or increase the dose, and you can keep the patches for a longer time. So it is going to decrease the dose of costly drugs. And further is like we are looking into the improving the designs where we want to put instead of needle kind of micro infusion machines, whereas like just like a saline drip, normal saline drip, this should be able to deliver the drug constantly through the skin. So these are the micro infusion drips. So this is our next aim, but like right now we are not working on it, but like we may work in future time. So there are several publications. I just cited a few of them are relevant to this presentation. And I would like to acknowledge my colleagues, Dr. Ajay Banga. He is my mentor and supervisor at Mercer University, Atlanta. Dr. Jairam Puram, he is my friend and colleague at Auburn University. And David Farquhar, he used to work for Altia Pharmaceutics. We used to work on industrial projects. He's the one who helped me with this MATLAB. Janice Baker helped me with this confocal microscopy from University of Nebraska. Lisa Hoskin is from Georgia Tech Hospital, and they have, it's a Georgia Tech University. They have a beautiful lab, and they have even capability to build a nuclear weapon with a short notice. So such as a very well equipped lab. So they helped me with the scanning electron and Hyrox imaging of the pictures. Dr. Lisa Hoskin, she's a director at that place. And Yashi Osaka, Dr. Yashi Osaka is he's a microneedle supplier from from Japan. He's the one who has his first microneedles, but we are the first ones to publish some literature on soluble microneedles. Thank you, everyone. And I truly appreciate the organizers for giving me an opportunity to present this. And
Chandra, if I may request,
yeah,
can you please for a moment on video and wave your hands to all of our audience?
yeah.
This-
I'm in my in my t-shirt. Can you just give me a second? I'll be there. Okay. Just 1 second. Sorry, it's quite early in California for us. So yeah. Please don't mind. I'm in my t-shirts.
Okay. Hello.
Yeah. So I had to unshare, right?
Yeah.
Unshare the screen. Stop
Yeah.
share.
Yeah. Start video.
Yes. Yes. Okay.
Yeah.
Thank you all. Thank you for giving me this opportunity to present. Oh my God, there are so many people. Yeah. It's nice to give a presentation to you all.
Yeah.
Yeah. If you have any questions, you can contact. I know there is a short of time, so my email address is with the organizers, and you can definitely contact me if I can.
Actually, Dr. Chandrasekhar, seeing you, and I'm really, really happy.
Hi. Yeah, likewise. Like it's been a while.
Yeah, almost 30 years.
Yeah,
And
yes, yes.
I thank you very much for accepting our request to be the speaker. And I thank you very much for enlightening our participants. And I have no hesitation in saying that, the session has been very fruitful. And the kind of explanation that you have given was very lucid, attractive. Starting from the very basics, you have slowly taken us to advanced issues like, from microneedles to micro infusion drips, which may be your future research area. And, the kind of definitely, we have definitely got benefited, we have got benefited by your lecture, content wise and quality wise. It was like, peeling off the skin and feeding the banana.
Yeah, that's right. Yeah.
Yeah, it was so informative and it was so interesting. And I once again thank you from the bottom of my heart for accepting our request in spite of your busy schedule. Okay.
It's my pleasure. It's
And
my
I'm
pleasure always.
yeah,
I'm fortunate because I was born in India. I was grown my primary education in India. My heart lives in India. And anything that I can do for India, no problem. I will be more than happy to.
yeah.
That's
So
my motherland. And
that's great to hear. Still, your Bhumi Putra, like that is your Karma Bhumi. This is your Janma Bhumi.
yeah, usually I visit India every year. So this time because of COVID, I was not able to, but usually I'll be there in summer every year. There. So
Yeah.
like to meet next time when I come there.
Yes. If I may request you in my personal capacity, please do not forget to give me information when you visit India next time.
No, certainly I will meet. Thank you. Thank you so much. Thanks for
Uh,
the invite.
yes. Yes.
Thank you all. Okay. Thank you
Thank
all
you.
for giving me this opportunity.
Thank you very
And wherever
one.
you are, good luck. Yeah.
Yeah. One more thing. Some of these participants may be having some doubts. I request the participants to send mails for clarifications, as we are going to have next session at about 9:30. So I may be, excused for not giving time for taking clarifications for your doubts. I request all the participants to bear with it and kindly cooperate with us. And you please please stay remain in the class, and next session is going to start at 9:30 or so. Once again, thanks very much to Dr. Chandra. Convey my best wishes to your family members.
Sure. Thank you all, sir.
Thank you. Yeah, now you can. Right.
Yeah, I'll log off.
Right, thank you.
Principal Sim.
Hello, Dr. Koti, are you there? Hello.
It's audible, sir. You can continue.
Namaste, Dr. Koteshwara, after a long time. Now I would like to introduce the speaker for next session. He is also known to me when I was in Malaysia. He was then working in Malaysia for Taylor University. So I take pleasure in introducing him to all the audience. His name is Koteshwar Rao, and we used to call him by name, Koti. He was my M.Pharm batchmate while I was pursuing pharmaceutical technology in Andhra University in the year 2000. 2000 passed out batch. He was in pharmacology class same year. And he has completed his B Pharmacy in the year 1998, which means 94 to 98. During these years, he has studied his B Pharmacy from Acharya Nagarjuna University, and then he has done his M Pharmacy in Andhra University from 1998 to 2000, and he has pursued his PhD once again in Acharya Nagarjuna University itself. He has completed successfully his PhD in the year 2007, and he worked for some organizations like Geetam Institute, and then he moved to Malaysia to further his career. And in Malaysia, he stayed from the year 2007 to 2016. He has worked there in a couple of colleges or universities. And from there, it seems like, he has moved to US, about which I really don't know. Now only I came to know. As the luck would have it, I am going to meet him after, say, 11 years. We used to frequently meet in Malaysia. I hope Dr. Koteshwar Rao remembers that, he used to come to Auntie Canteen for having dosa. So I still remember those memories which are vivid in my mind. And he looks very simple, but he's highly knowledgeable. And if you look at his career, we can easily understand how progressively he has moved from one position to another position. It's worth emulating. Presently, he's working for California Health University as assistant professor. And I once again take great pleasure in inviting for second day of this webinar. Now I request Dr. Koteshwar Rao to kindly take up the session and enlighten us. Thank you very much. Dr. Koteshwar Rao, please go ahead. Hello.
Sir, speaker's voice is very low. Can you increase, please, your volume? It's not audible, sir, still. Hello, sir, can you hear the voice? It's not audible to us.
Dr. Surya sir, can you please call him and tell that we are not able to hear?
Sir left, sir. Maybe he is having some technical issue.
Okay, let's
Wait for 5 minutes.
yeah, let's wait, and I request the participants to be please
Sir, join. We can check it now.
join. Okay.
Can you hear me?
Yes, sir. You can continue.
Oh, okay. Now it is clear.
Yes.
So how can I share the
I'll
screen?
stop sharing here. Then you can proceed with that.
All right.
Now you can continue, sir.
Okay. Can you see me now?
Yes,
See my
sir.
screen?
Yes, sir.
Hi, good evening, everyone. Thanks, Ramana, for the introduction. My name is Koteswar Rao Nalamalu. I'm going to present inflammation role in post-stroke brain damage and neurological functions. I'm an assistant professor in California Health Sciences University. Before coming here, I worked in University of Illinois. This work is done mostly in University of Illinois, and some part of revisions we did in California Health Sciences University. So before starting inflammation, I'd like to talk about stroke, why we have to conduct research in stroke. Stroke is a sudden interruption of blood flow or a rupture of blood vessel. It is actually a cerebrovascular accident. When it occurs, we need to take care immediately. There are two types of strokes. One is ischemic stroke, another one is hemorrhagic stroke. Ischemic stroke, mainly due to a major clot formed by blood clotting or an atherosclerotic plaque obstructed the major vessel in brain. So due to that, there will not be further blood flow to other regions. This is called as ischemic stroke. Hemorrhagic stroke is blood vessel ruptures and leaks the blood to adjacent regions of blood vessels, brain cells. And the brain cells will become hypoxic and hypoglycemic in both conditions. When brain cells become hypoxic and hypoglycemic, many mechanisms evoke, tries to compensate the situation. Instead, it causes more and more damage to the brain cells. So we have to see what are the symptoms. When someone got face drooping, arm weakness, or speech difficulty, all of them suddenly together, call emergency, immediately enter the patient into a hospital and get treated as soon as possible. Why we have to be concerned with stroke? Stroke is the second leading cause of death in the world. 1 out of 20 patients deaths due to stroke in the United States. And it's the leading cause of long-term disability. Long-term disability is mainly due to ischemic stroke. Long-term disability causes many problems. The person, if he is the breadwinner to the family, he can't earn the money, he can't work. So total family will be under distress. Again, this disabled person need a caregiver. Depending on the intensity of disability, a caregiver need to take care of the patient from few hours to the whole day long. This is another burden to the family again. It needs to be addressed. Every 40 seconds, a stroke case is reported. Every 4 minutes, someone dies of stroke. Over 600,000 stroke cases reported every year. And as I told, economic burden is increasing year by year. It was over $70 billion in 2012 and is projected $180 billion by 2030. And among these two types of strokes, ischemic stroke and hemorrhagic stroke, 87% of the strokes are ischemic strokes. So in my research, we did research in mainly we concentrated on ischemic strokes. Reason behind that is ischemic strokes are major contributors of long-term disability, and patient survival is, majority of the cases, patient survival is possible. Whereas in ischemic strokes, fatality rate is very high, over 90%. And long-term disability, in that 10% cases, severe long-term disability is observed. So we wanted to work on ischemic stroke. We wanted to prevent the ongoing brain damage and retrieve back the cells, improve the neurological functions. That is our aim in our research lab. So when somebody got stroke, time is important. Every minute counts. Every minute's number of brain cells will be dying more and more. We have to act fast. We have to treat the patient immediately. Otherwise, the disability intensity will go on and increase as it progresses. Time is wasted. Intensity will be worst. Stroke outcome will be more worst in terms of disability terms. So when stroke occurs due to either thromboembolic plaque or atherosclerotic plaque, some brain cells at that particular region will die. We call them as core, and the core is already in this region. The cells are died. And the adjacent region to the core is called as penumbra. Penumbra is the place where the cells are under crisis now. If time is delayed, treatment is delayed, then the penumbra also starts, the cells in that penumbra also starts dying, and the stroke outcome will be more worst. So what is the treatment approved till now available? Tissue plasminogen activator, known as TPA, is approved drug by FDA. This is the only drug available. What does TPA do? TPA converts plasminogen to plasmin, and this plasmin cleaves the fibrin-induced clot into fibrin degradation products. So the clot is cleaved. The blood vessel is free. Now blood can flow. So reinstate the blood flow. We call it as reperfusion. First phase, where there is no blood flow and no sufficient oxygen and glucose supply, we call it as ischemia. When we cleave the clot, reinstated the blood flow, we call it as reperfusion. Because I'm going to talk about them in my subsequent slides. Remember ischemia and reperfusion. So how long we can treat this TPA? Is it like after 2 days, if the patient enters into the emergency department, can we treat? No. Within 4.5 hours of stroke onset, the patient must be in emergency department and must get treated with TPA. Then somebody asked me, what happens if we delay the treatment? If we inject a TPA after 5 hours, 6 hours, 10 hours, 12 hours, what happens? It causes hemorrhagic stroke. It ruptures the blood vessels. After a few hours, for 4.5 to 5 hours, if we inject TPA induces capillary permeability and leaks the blood and causes hemorrhagic stroke, which is more fatal. Recently, I heard about a patient when I was talking to a doctor, give him TPA within 4.5 hours. And doctor told, sorry, we can't, because patient entered after 12 hours, it's not possible to treat with TPA. If that patient is given with administered TPA, that might have caused much more worst outcome, maybe fatality. That's the problem. So why we need novel therapeutics? TPA only cleaves the clot. It won't address any other things, just induce reperfusion. It doesn't control the ongoing brain damage. It doesn't address the secondary brain damage caused due to inflammation. It doesn't retrieve any lost neurons, brain cells. Only thing TPA can do, it will reinstate the blood flow and at least supply oxygen to the live cells, which are still alive, will get oxygen and glucose. Finally, the stroke, ischemic stroke, led to motor and sensory functions loss due to damage in cortex region and many other regions. TPA won't address that. TPA can't retrieve any neurological functions. So we selected, we are not targeting everything with this mechanism, because stroke evokes, as I told, lots of mechanisms which are detrimental to brain cells, neurons. So we need a combination of many molecules. We worked on MMP12, we worked on stem cell therapy, exosome therapy. One more target for us is acute inflammation. Acute inflammation mainly targeting the penumbra region, which is adjacent to the dead tissue, which is under heavy crisis. If we can't prevent the ongoing damage, the penumbra also will be damaged and the core size will be increased rapidly. That's the problem. So we studied the literature. What happens when acute inflammation is on song? What happens in the brain? Acute inflammation activates the resident microglia, astrocytes, reactive oxygen species are released, nitrogen species, inflammatory cytokines are upregulated. Altogether, we call it acute inflammation is at its level best. So this acute inflammation promotes the secondary brain damage and starts inducing apoptosis to the penumbra region brain cells. And we further studied, is there a particular target which is inducing all these processes? We found that toll-like receptors play a key role. There are many toll-like receptors. We wanted to target the major contributors. We don't want to target every toll-like receptors which are having different structures. We found in literature TLR2, TLR4 are contributing major in stroke-induced penumbra damage. So what happens, we further studied what is this TLR2, TLR4 doing? What's the detrimental signaling pathway? TLR2, TLR4 evokes the MYD88 dependent pathway, and MYD88 activation induces IRAX and TRAP6, then induces the nuclear factor kappa B transcription, and nuclear factor kappa B enters into the nucleus and upregulates the release of TNF alpha, IL-1 beta, and IL-6. TNF alpha, IL-1 beta, IL-6 are inflammatory mediators. They cause ultimately cell death. Then we also want to know whether this TLR mediated damage is from peripheral region or in brain. Is there any expression of TLRs 2 and 4 in brain? We found that microglia, astrocytes, oligodendrocytes, and neurons contains TLR2 and TLR4 expression. All right, great. We found that literature had shown a way to us. If we can control these areas, TLR mediated activities, we may able to control the damage. Then we further studied literature, what others found. There was some confusion on TLR2 role. Somebody told that TLR2 is having some role, protective role compared to wild type rats. They told that TLR2 is neuroprotective. TLR2 is detrimental to neurons. TLR4 is neuroprotective in one study. Another study indicated that TLR2 inhibitor bicalin could be neuroprotective. So some reports shown that TLR2 is detrimental. Some reports shown that TLR2 is neuroprotective. There was a confusion in TLR2 aspect. And another study also shown that TLR2 suppression by some TLR2 knockouts, they tested in TLR2 knockouts induced ischemia. They found that there is a significant reduction in infarct size. And in TLR4, there is no confusion. TLR4's inhibition can be neuroprotective. They reported it. So what is our role in this? What we did? First, we want to get rid of the confusion due to TLR2. Second, we wanted to suppress simultaneously TLR2 and TLR4. And we want to test if we can suppress simultaneously both these. Can this able to prevent the ongoing brain damage? Can this able to protect the neurological or retrieve back or prevent the neurological functions? That was our hypothesis. And this article is published in Neuroscience. Another part of this talk is already in communication with translational stroke research. We selected rat as a model. We chose middle cerebral artery occlusion, and middle cerebral artery occlusion is a microscopic surgery where you need to expose the common carotid artery and external carotid artery. We clamp the common carotid artery with the arterial clamp to prevent the blood flow to further. We clamp, we made a tight knot at one end of the external carotid artery and a loose knot at another end of the external carotid artery. And we also we also exposed the internal carotid artery and placed an arterial clamp to prevent the blood flow here. And this tight knot prevents the blood flow to this region. And we made an incision here. We inserted a monofilament silicone rubber coated filament through this, and that monofilament traversed from external carotid artery via internal carotid artery and reached the middle cerebral artery. So here we are blocking the middle cerebral artery by introducing a filament, monosuture, silicon rubber coated monofilament suture. So blood flow is blocked here. It mimics thromboembolic or atherosclerotic plaque induced clot, obstruction of the blood flow. So we, in real picture, it looks like this. These are microscope, taken from microscope view. And we introduce, this is the internal carotid artery, external carotid, and common carotid artery. Through the external carotid artery, the loose knot made and introduced the filament, and then it was tied. After introducing the filament, we tightened the knot, and we took all the clamps, arterial clamps, we stapled it, and this surgery takes only 10 to 15 minutes by, which is a really difficult surgery. We need consistency. We need a rat alive. We anesthetized the rat using isoflurane, 2 to 5% isoflurane continuously. So we anesthetized the rat, we performed the surgery. It takes 10 minutes. After surgery is over, the common carotid artery clamps and internal carotid artery clamps are taken out and reinstated the blood flow, but only at the meridial cerebral artery region, there is no blood flow. And we staple the skin of the rat and we allow it to stay for 2 hours. During these 2 hours, there is a blockade. So 2 hours of ischemia induced. After 2 hours of ischemia, we reopened the rat and we took out the filament and tightened this knot again after taking out the filament. And we sutured back the rat skin at the neck region, and the rat is still alive. After doing this experiment, somebody will question, hey, is there a brain damage? Yes, we performed TTC. TTC stain, when it enters into the brain cells, due to the presence of enzymes, the TTC will be converted into, in a live cell I'm talking about, in a live cell, the enzymes will convert TTC into parmesan. Parmesan is an insoluble pigment which stains the live brain cells. And there is no live cells. The TTC can't be converted into parmesan and it can't stain. See here, when we induced post-stroke, some part of this is a ipsilateral region. This is contralateral region. We induced stroke in ipsilateral region. So there is so much brain cells death you can see here. So there is a brain damage. But we can't do, every time we can't sacrifice the rat, we can't test whether there is brain damage or not. This is not feasible because we need a live rat for 2 days, 3 days, or 14 days, 20 days, depending on our experimental requirements. We need live rat with stroke. So we introduced another test. One of the tests we do is open field. A normal rat can walk freely straight to normal. When a stroke-induced rat is there, because we induced stroke at the right side, the left side paralysis will be more observed, and rat will be circling towards its towards one kind of circulation it will do. It can't able to do circling on both sides. One side only, it will be continuously circling. And if you hold the tail of the rat, normal rat paths will be very parallel, similar and parallel to each other. But in case of stroke-induced rat, the injured or disability-induced forelimb can't able to extend. Only one limb can be extended. It tries to catch the platform with one forelimb, but the other forelimb can't be extended. Third is beam walk. When we place it on beam, because we train the rat before surgery to walk on the beam continuously from one end to another end. A normal rat can traverse through freely, whereas stroke-induced rat, because of its disability, there is no grip strength and motor coordination. It can't able to stay on the beam. Even if it stay on the beam, it just hugs the beam, can't able to traverse through. So if we confirm this kind of tests, all are passed, then we consider the animal into the experiment. Otherwise, if the animal fails to show stroke-induced symptoms, we remove the test, we remove the animal from the study. We exclude that animal. So we collected brains after stroke, after reperfusion. Immediately after reperfusion, we sacrificed 6 animals. We collected the brains, and immediately after 6 hours of reperfusion, 12 hours of reperfusion, we collected the brains and we extracted the RNAs using trizol method, and we tested for expression of TLRs. We found that TLRs are significantly upregulated. This is mRNA expression. We ran RT-PCR and agarose gel electrophoresis. TLR2 and TLR4 upregulation is observed from 6 hours itself. 12 hours also we could see marked upregulation. And we wanted to further see whether there is this upregulation is continued further. We tested on day 1. We sacrificed 2 hours of ischemia, 1 day reperfusion rats. We sacrificed 3-day reperfusion, 5-day reperfusion. We sacrificed 6 rats per group in each cohort, and we ran whatever experiment from here I'll be talking. We collect the ipsilateral lobe of the brain because contralateral doesn't have any stroke. So ipsilateral lobe of the rat brain is collected, isolated RNAs, and then we ran a real-time PCR for TLR2 and TLR4. We could see there is a significant upregulation on day 3, day 5 compared to the normal animal. So TLR2 and TLR4 are upregulated. It's confirmed. Then we want to see whether it is translated to the downstream signaling molecules or not. We tested, we ran RT-PCR. We also ran Western blots by collecting other cohort of group of rat brains, ipsilateral rat brains. We prepared cell lysates using RIPA buffer, which is a strong buffer which can extract the nuclear contents as well. With protease cocktail inhibitor added to that, we prepared the cell lysates and we ran Western blots. In mRNA expression, we ran RT-PCR, we ran Western blots. In both cases, there is a upregulation of MYD88, IRAK1, NF-kappa B in day 3, day 5 of the post-stroke induced animals. We selected beta-actin and GAPDH as the housekeeping gene. And we also wanted to see whether it is also seen in inflammatory mediators. We could see mRNA expression of inflammatory mediators and the protein expression of inflammatory mediators. This is RT-PCR, this is Western blot. IL-1 beta, IL-6, TNF alpha are upregulated until 5 days of reperfusion. So we wanted to reduce, our aim is, as our hypothesis says, our aim is to reduce the intense inflammation. How we can do that? Brain is inflamed right now, but not both parts. The ipsilateral region is inflamed because we induced stroke in one region only. How to reduce intense inflammation? We wanted to suppress specifically TLR2 and TLR4. How we can achieve that? How? By administering shRNA plasmids, which are embodied in E. coli. And these shRNA plasmids are nothing but short pin, hairpin RNAs. They will be introduced into the brain and they will knock down the targeted gene. The vendor supplied 4 clones for each TLR and TLR2 and TLR4. Are we going to work on 4 clones? If I work on 4 clones, I need a large number of rats. My boss will kick me out of the lab. So what happened is, what I did is, I searched, is there any cells which can express TLR2 and TLR4? We found that pheochromocytoma, PC12 cells, highly expresses TLR2 and TLR4. After the shRNA plasmids are synthesized and purified, synthesized using Quigen Maxi Prep Kit and purified using phenyl chloroform isoamyl alcohol. Then that purified plasmids, I cultured PC12 cells. I transfected the TLR2 and TLR4 using Fusion reagent. So TLR2, different clones are transfected in different plates of PC12 cells. TLR4 is clones are transfected in different plates. Different clones are transfected in different plates. We found that every clone is working, but one of the clones, clone 2, we just named it as clone 2. Among the 4 clones they provided, both are working highly efficient. So this efficiency attracted us, and we selected these clones for further study. And we injected those TLR2 shRNA, TLR4 shRNA, clone 2 selected by us through in vitro studies. We injected to post-stroke rats. Immediately after reperfusion, we added a nucleic acid delivery reagent called in vivo jet pay reagent, in vivo jet pay reagent, added mixed with TLR2 and TLR4 independently. We prepared a formulation and we injected intravenously to the rats after 2 hours of ischemia. Immediately after reperfusion, after taking the filament, we injected this formulation. And we found that in untreated and vehicle treated, this is a scramble vector control. In vehicle treated, there is no significant changes in TLR2. Still they are upregulated. But TLR2, TLR4 injected, shRNA injected animals, there is downregulation of TLR2 and TLR4. Then we were very happy, in vitro and in vivo, these plasmids are efficient. We wanted to see whether it is translated to brain damage or not. And we did TTC for sham, which is just we operated. We haven't introduced any filament to the rat brain. It is just operated and sutured back. And untreated, we introduced the filament, induced 2 hours of ischemia. We sacrificed this group of rats after 1 day. We injected only TLR2 shRNA, and because we wanted to see whether it is both in combination or only one of the shRNA is producing the effect. We have to differentiate it. So we injected TLR2 shRNA to one cohort of rats, TLR4 shRNA to another cohort of rats, and a combination of TLR2 shRNA and TLR4 shRNA to another cohort of rats. Compared to untreated, if you compare sham with untreated, there is a significant damage. But when we injected TLR2 alone, shRNA alone, TLR4 shRNA alone, we could see there is a significant drop in infarction, brain swelling. And when we gave together TLR2 and TLR4 shRNA together through intravenous route, we could see that there is a significant reduction because this prevented the secondary damage. So we could see there is a marked improvement, marked difference in comparison with untreated rats. So brain infarct size is reduced significantly, and swelling also reduced significantly when we suppress together TLR2 and TLR4. And we wanted to see whether it is also translated through downstream signaling molecules. We could find that MYD88, NF-kappa B, and inflammatory mediators all are downregulated. We tested them after 3 days. So we collected the ipsilateral lobes of the brains after treatment 3 days later. We compared with 3 days untreated rats with sham. So sham obviously will not show major expressions. Untreated rats, there was heavy expression of downstream signaling molecules and inflammatory mediators. Also, when we treated with TLR2 shRNA and TLR4 shRNA together, we could see that there is a downregulation of the target molecules. Well. We could see that the brain damage is reduced, inflammation is reduced. So we don't want to stop here. Inflammation, we haven't achieved a final target. We wanted to see whether this mechanistic approach is translated to neurological functions or not. So we did neurological evaluations. Before surgery, from here, whatever group of animals I will be talking, that group is N is equals to 10. Previously, I was talking N is equals to 6. You please take note of it. So what we did is we trained the rats before a surgery. We trained the rats for 3 days, and for beam walk, sticky tape, rotarod like tests. I'll be discussing what are those tests, how we did it later. So we trained the rats for 3 days, and those rats which performed to our benchmark standard are included in the study. If any rat fails to perform, that is excluded from the study directly. So after they reach to 220 to 240 grams of the body weight, 6 to 8 week old, they are given the trainings before surgery. And on the day, we induced middle cerebral artery occlusion, and the ischemia was induced for 2 hours. After 2 hours, we took out the filament and reinstated the reperfusion blood flow. And after this, immediately after reperfusion, sorry, this is not showing the exact one. After 1 day, we have injected the what we call it as TLR2 shRNA and TLR4 shRNA. And then we tested until day 7, day 14 accordingly. So we do, after injecting the plasmids, post-surgical care, we tested them after 2 hours of ischemia. We tested them for MNSS tests. If they don't show MNSS score of 9 to 12, 8 to 12, we excluded it. So what is MNSS test? Modified Neurological Severity Score Test. It is a series of battery of tests which test the motor, sensory, and many other functions. MNSS test scoring ranges from 0 to 18. 0 indicates there is no disability. If 18, severe disability. 1 to 6 is mild stroke. 7 to 12 is moderate stroke, and 13 to 18 is severe stroke. We don't want severe stroke induced rats because mortality maintenance is very, very difficult. They die on the slot. We want the animal to be alive for 7 days or 14 days because we want to test their improvement. So we selected moderate stroke induced rats. And what we did, I will be showing you some tests. One of the tests, if you see, stroke-induced rat will be circling like this. If it is circling, we will score it 2. If it is unable to walk straight, we will score it 1. If it is falling to a paretic side, we will score 3. We don't consider falling to paretic side rats because that is severe stroke. So those rats which are circling, we will score 2 points for that. Another test we will do is balancing on the beam. See here, when we induce stroke, the rat forelimb and hind limb disability is clearly seen in this test. If the rat falls down immediately, we will score it 6. If the rat falls after before 15 seconds to 20 seconds, we will score 5. Like that, this score ranges from 0 to 6. 0 is easily able to walk from one end to another end, but 6 is whenever we put it on the beam, unable to cope up with the balance and no grip strength, so it will fall down. So we will score it as 6. So 6 is worst and 0 is great, no neurological disability. So here, these two tests differentiates of normal to stroke-induced rats and to treated rats. Another one is another test is sensory test. So in sensory test, we touch the vibrissae of the rat. The whiskers will be touched to an object, and the limb is freely suspending in the air. Whenever it touches, we touch the vibrissae, normal rat can place the limb on the table, whereas stroke-induced rat will not be able to place the forelimb on the platform. So that disability, we will score it as, if there is disability, we will give a score of 1. Like that, we will be doing a series of tests, how it is walking, how it is on the beam, how it is performing in the placement test, vibrissae, when we touch the vibrissae, can it able to place it properly? All this together, the entire score is 18, 0 to 18 ranges from. So when we induced stroke on day 1, in both cohorts, there is a significant increase in MNSS score, around 9. We could see that this rats are having great disability. So consistent disability was seen on day 1, but the disability was sustained more in untreated rats or vehicle-treated rats. Whereas in TLR2 and TLR4 together treated rats, the disability was rapidly decreased. And that means neurological functions are improved. You can see on day 5, there is a significant difference between the vehicle treated rat and TLR2, TLR4 together treated rat. So significant difference. This difference we can see there is a improvement over 14 days period. If you see from day 5, there is a rapid improvement. You may ask me, there is also improvement in control group. Rats have the capability, because these are young adult rats, they have capability of self-recovery. But in case of TLR2, TLR4 shRNA treated rats, this improvement was seen significantly. So through this, we identified that motor sensory functions are improved in TLR2 and TLR4 treated shRNA treated rats. That means by suppressing TLR2 and TLR4, we could see some improvement. We didn't stop there. We also did beam walk test. Beam walk test is nothing but we will be training rats before surgery to walk on 1 meter beam from one end to another end freely. This rat is stroke-induced rat due to its motor incoordination. It doesn't have proper motor coordination, and it doesn't have grip strength. So it couldn't able to stand on the beam. So those rats which can't able to stand on the beam will be scored 0. If they stand on the beam, hugs the beam, will be scored 1. If there is some foot slips more than 50%, or if it starts moving, tries to move, traverse through but fall down, will be scored 2. Like that, we will score 6. 6 is worst when it 6 is the best. When it is able to travel from one end to another end freely, we will score it 6. So 0 is worst, 6 is the best. So here in this video, if you see it again, the rat is not able to withstand on the beam, and it could fall immediately, very easily. Could you see that? See, there is a great disability in its limb, hind limb, as well as forelimb also. With one limb only, it can able to catch the beam, but the other limb unable. So it is the rat, we scored it 0. Like that, we did this test over a period of 14 days again. On day 1, we could see there is no significant difference. After induction of stroke, both the rat groups are having similar kind of stroke. But there is a little bit of improvement because we injected immediately TLR2 and TLR4 shRNA. After 24 hours, there is a little improvement, and as it goes on, there is a little bit rapid movement improvement compared to the vehicle control. And there is a significant difference on day 14. If you see the star, there is a significant difference between the vehicle treated as well as TLR2, TLR4 shRNA treated. Even though there is no much difference, but we can see rapid improvement in TLR2 and TLR4 shRNA treated cohorts. So 14 days this test could show that there is an improvement in motor coordination of the rat. And this is a sensory test. We call it as adhesive removal test. We will place a tape on the paw, cover the paw, and we will allow the rat freely to walk on the ground on a platform. This is a nice clean tub. So what we will do is we will place the tape, and we will on the 2 timers we will have. We will after when it is acclimatized and first starts attempting to remove the tape, we will start the timers at that time, and we will count 30 seconds. One timer will be continuously running. The other timer will count the seconds of attempts. Whenever it is attempting, we will on the timer, the second timer. Whenever it is not attempting, this timer will be off. So when it reaches 30 seconds, this timer will be off as well. And we will see in 30 seconds how many seconds it is attempting to remove the tape. You can see here, this is the normal rat. See, it is attempting continuously to remove the tape. The tape is obstructing the paw. So through walk, the sensory coordination is there. It could able to identify and remove the tape easily. This is about normal rat. But what about a stroke-induced rat? Stroke-induced rat, it won't be attempting. See, even the tape is surrounding the paw. Due to lack of sensory functions, it lost the sensory function. It couldn't able to attempt to remove the tape. See, this is running. The first one will be running. Second one will be active only when it is attempting. When 30 seconds is completed, this we will stop it, and we will measure how long this rat attempted and we will note it. We will count we will do this test for affected limb and unaffected limb. We will take the ratio, sticky tape ratio of affected limb divided by unaffected limb, number of the ratio. And we found that the attempts were very low on day 1 in both stroke-induced vehicle treated rats as well as stroke-induced TLR2 and TLR4 treated rats. This indicates in both cases stroke is significant. In 7th day and 14th day, we can see a marked improvement On 14th day, there is a significant difference between the vehicle treated as well as TLR2, TLR4 SH treated rats. So we can conclude here that sensory functions are improved. And the last test is rotor rod test. Rotor rod test evaluates the grip strength and coordination. What happens is this rotor rod starts at 10 RPM, and every second it will improve 0.3 RPM, and it reaches to 40 RPM, that's the maximum. So see here, the rotor rod starts, the rat will be holding according to their ability, even though there is improvement in speed. So the disabled rat fall down here. The normal rat are improved neurological functions due to TLR4, TLR2 treated rat will stand more time compared with the untreated or vehicle treated rat. That is rotor rod test. Rotor rod test mainly test the balance, its grip strength, coordination, everything on the accelerating rotor rod. When we tested it, we could see that there is a difference between these two. On day one itself, the treated rats could show some improvement, and there is a marked difference on day 14. As you see, the recovery of neurological functions. Why? Because we suppressed the acute inflammation. There is no further damage. So improvement is more rapid compared with the vehicle treated rats. That's the difference here. So in conclusions, TLR2 and TLR4 expression was upregulated after focal cerebral ischemia in our MCAO model. And simultaneous suppression of TLR2 and TLR4 reduced the infarct volume as well as brain swelling, and also downregulated the pro-inflammatory cytokines, improved neurological functions. So we tested from top to bottom. We tested mechanistically. We also saw whether it is translated into neurological functions improvement or not. So we found everything is all right. So now we are targeting the MyD88 pathway. We confirmed MyD88 presence, MyD88 detrimental role. We are targeting MyD88 right now. I want to acknowledge my friend, mentor, Krishna. And also would like to thank Dr. Bharat Cheluboina, who is now a research scientist in Wisconsin-Madison, and Dr. Nathan Smith, who was with me. I was his mentor. Wherever he went wrong, I used to screw and set him right. He was very happy to work with me. Dr. Jeffrey Klopfenstein, neurosurgery, head of the neurosurgery in our university hospital, who is our collaborator, and continuous support from him. Dr. David Wang, head of neurology in our hospital. Dr. David Pinson, pathology specialist, Siva Reddy, and Dr. Casey, my colleagues, Natalia, Lara, and Maunib. They are the blinded evaluators for neurological functions. We did blinded evaluations. So I would like to thank everyone. I would like to thank organizers for giving me this opportunity. Ramna, thank you. I saw you after a long time. It may be so many years. Yeah. Thanks to everyone for attending this session.
Just a moment, please.
Any questions?
Yeah, Dr. Koti, I thank you very much for your nice presentation of your work done. And I can say comfortably and confidently that even a non-pharmacology person can understand your class easily. and the presentation was so conspicuous, and it was very informative. And what attracted everybody's attention was, your visuals of
Thank you, Ramana.
the animals in vivo studies. That has really, grabbed everybody's attention, because seeing is believing. And I, as a teacher, of course, we as teachers understand that how much expertise it takes to execute work on small sized animals like rats. One has to have more accuracy, precision, and expertise. Then only we can execute the work as per the plan. And it shows that how much work that you must have put in to get the results. And it's really informative and beneficial to all of us. And your presentation was simply superb. And I once again thank you very much for your lecture. And once again, I thank you for accepting our invitation in spite of your busy schedule and sparing your valuable time. With this,
Thank you very much, Ramana. Thank you. One girl was talking, I don't know her name. Thank you to her also. Initially, there was some problem. It was rectified immediately. Thank you. Thank you so much.
Thank you all. And just a message I want to pass on to the viewers, that tomorrow is the last day, and some requests were coming to provide feedback forms. By tomorrow morning, we'll be able to provide feedback forms, and your feedback is very important for us. And I thank every participant for the patient hearing and the interest shown to attend the seminars. And I thank everyone once again, and good night.
Good night to you all. Bye.
Sir, session will be closed now. Sir, can you hear?
I can hear.
Sir, I'll close the session now.
Yes, we can close.